Greetings, everyone. Welcome to our new webinar series in partnership with the Montana Natural History Center and the University of Montana, The Age of the Coronapocene. I'm Jennifer Robinson, Education and Program Director for the Montana Natural History Center, and will be your host for this exciting new series on the natural history and science behind the virus COVID-19. Drew Lefebvre, Montana Natural History Center Museum Programs Coordinator, will be your co-host and working in the background to answer your questions and address comments throughout the program. This webinar is supported by closed caption capabilities. The wonderful Caroline Habish will be working in the background to provide text for our speech if you need assistance in following along with the program. Please click on the link in the chat box that Drew will leave there. This will open up a separate window where you'll be able to read what is being said on screen as we are speaking. Please note that the view cap for the closed caption is only 50 individuals. Please only use this assistance if you need it, so we have enough space for all who require closed caption assistance. Throughout the webinar, we will repost this link into the chat box for those who join late. Joining me today are a panel of incredible professors, scientists, and educators from the University of Montana. Please note that our panel are not medical doctors and will not attempt or try to diagnose the symptoms or any of your symptoms in this program of COVID-19. Later in the program, the audience will have an opportunity to ask our panel questions about the virus and living with COVID-19. But note that we will not be taking questions seeking medical advice. Please seek medical help if you have questions or concerns about your own medical health. So I'm going to jump in and introduce our panel of scientists and educators. First, I'd like to introduce Professor Erick Greene. Professor Greene works in the Division of Biological Sciences and Wildlife Biology program at the University of Montana. Dr. Greene is an ecologist who studies complex interactions in biological systems. He has broad interests in behavioral ecology, evolutionary biology, and conservation biology. Next, we have Assistant Professor Angela Luis. Professor Luis works in population and disease ecology at the University of Montana and studies the epidemiology of infectious diseases. She is an expert on how diseases spread between species, between individuals within a species, and the dynamics of infectious disease. Finally, I'm thrilled to also introduce you to Associate Professor Rosalyn LaPier. Rosalyn works in the Environmental Studies Department at the University of Montana and is an award-winning Indigenous writer, ethnobotanist, and environmental activist with a BA in physics and a PhD in environmental history. She works to strengthen traditional ecological knowledge and revitalize Indigenous languages within Indigenous communities. Dr. LaPier is an enrolled member of the Blackfeet Tribe of Montana and Métis. We have one more panelist that will be joining us in next week's webinar for this series. Brandon Cooper, Assistant Professor in the Division of Biological Sciences at the University of Montana, is a microbiologist who studies evolution of bacteria. He also conducts research about how some important human diseases are transmitted and spread. The purpose of this webinar series is to understand the science behind the novel coronavirus, COVID-19, and discuss how it affects human health and society. With all the information swirling out there about this virus, we want to provide some clarity and understanding about the facts and give you, the audience, the chance to hear expert scientists discuss these topics and answer your questions. In today's episode of the series, I will ask each panelist a question to start a discussion about how their area of expertise relates to the science behind COVID-19. This will take up about half the time allotted for the webinar. After each panelist has shared, we will open it up to the audience to ask questions. To ask your questions, you're going to be using the question and answer feature in your Zoom app. In your Zoom toolbar, look for the icon that says Q&A. Click on that icon and type in your question. Only panelists will be able to see your questions, and if accepted, we can share it with the whole webinar. Depending on how much time we have and how many questions are asked, we might not get to all questions, but we will save and review them to help inform content for future webinars in this series. If you'd like to leave comments or let us know of any technical difficulties during the webinar, you can use the chat box. Only panelists and hosts will be able to see your comments, but that's a good way to communicate to us if you need anything. Let's go ahead and try using the chat box now to make sure that it works for everyone. Click on the chat icon in your Zoom toolbar and tell us where you're tuning in from so we can see where everyone is watching from today. Looks like we got some folks from Missoula, Florence, British Columbia, Chicago. Nice, Patty Canyon. We've got Reno, Nevada. Wonderful. Yakima, Washington. More from local here in Missoula. Great. Some more from Missoula. Corvallis. Awesome. Utah, or Moab, Utah. Nice. I combined those two together there. We've got some folks from a long ways away in different places. This is great. So if you have any questions or comments during the webinar, please leave those, you can put those right into the chat box and we'll be able to reach out directly to you and address those during the webinar. Now we're going to turn to our panelists and get the discussion started. We'll begin with Professor Greene. Professor Greene, can you tell us what is a virus? And within that explanation, what exactly is novel coronavirus COVID-19?
Thank you, Jennifer. I'm going to share my screen right now, and I've got a little presentation. We mean to keep these really conversational and informal, and but I've got a quick presentation. Here we go. Make sure I can get it up and running. All right, do you folks see that? Can you see that, Jennifer?
Yes, I can.
Okay, so good afternoon, everybody. We hope that you, your friends, and your loved ones are healthy and safe during these really stressful times. And I'm going to give a really short presentation about the natural history and biology and viruses. And we hope that this information helps you understand a little bit more about viruses, what they are, how they work, and why they're fascinating but also scary. And we hope that these webinars help you understand more about COVID-19, how pandemics work, and what we can do about this one, and what the future holds. All right, so I'm just going to start with some basic natural history of viruses. So the first point I want to make is that viruses are really small. So here's some cartoons of a generic human cell, a bacterial cell, and a coronavirus. But I've shown these pictures all about the same size. But if we were to scale the images so they're about the right sizes relative to each other, you can see that the viruses, it's that little dot there, that little tiny little point. It's much smaller than bacteria, and it's thousands of times smaller than any one of the cells in your body. And in this microscope picture, you can see just how small each coronavirus is. These little dark dots are relative to this host cell. Okay, just to give you an idea of how small they are. The other thing that is important to realize is viruses are really, really abundant. And to help you calibrate this, a 5-gallon bucket of ocean water contains about 200 billion, that's with a B, viruses. And this is mind-boggling. This is about twice the number of stars estimated to be in the universe. So viruses are everywhere, and they infect just about every known species of animal, plant, and bacteria. And viruses are a really diverse group. Biologists put them into 7 different groups according to how their genetic information is stored. So coronaviruses shown here in panel B belong to what biologists call the group 4 of the virus family. And just technically that means that they store their genetic information on a single strand of RNA. So these are the coronaviruses that we hear so much about, and we'll be spending the rest of the webinars on these. And all animals, plants, and bacteria, they all have the genetic information and the cellular machinery they need to grow, copy their own DNA, and reproduce. But viruses are very, very different. And although they do contain small amounts of genetic material, it's not enough for them to grow or reproduce on their own. So this is what I mean when I say that viruses are the ultimate parasites. They depend entirely on the host cells that they invade to make new viruses. And here's a representation of a coronavirus. And they get their name from these surface knobs. So corona means crown. And these crown knobs, so this is where they get their name. These little knobs look a little bit like crowns. So these crown knobs help the viruses get into the host cells, and the genetic information is packed inside of here. All right, this cartoon summarizes the life cycle of all viruses, no matter what type they are or what their host is. And the first really important step shown over here is for the virus to gain admittance into the host cell. Now, cells don't like foreign things getting inside them, and they make it very hard to do so. So the virus's first trick and the first challenge they face is right here, is how to fool the host cell into letting them inside. And this is what the function of the surface knobs, or the corona, the crown of the coronavirus are. These knobs are able to attach to receptors on the surface of the host cell and fool them into letting them get in. And in this way, viruses are very much like the Trojan horse, which was used by the Greeks to fool the citizens of Troy. So the people of Troy brought the Trojan horse inside the defensive walls of the city, much like a cell would bring in a virus inside. And at night, the Greek soldiers jumped out of the horse and destroyed the city. All right, so once the virus gets inside of the host cell, the viral DNA completely hijacks the genetic machinery of the host cell. And so here, the virus in this cartoon has been successfully admitted. It releases its genetic information, and then here's where it completely takes over the genetic machinery, and it basically turns the host cell into a gigantic genetic photocopy machine. And so it gets the host cell to do the work of copying, making new viruses. And it's rather amazing, astounding that just one virus getting into a host cell and hijacking the situation and creating this genetic photocopy machine, one virus can be turned into 1 million new viruses. So you can see how potent this is. And so for when we're talking about corona or COVID-19, these are the droplets that would be released, say, from our lung cells, and would be coughed or sneezed out. And Angie and others will be talking a little bit more about that and transmission. All right, as an example of just how little genetic information is carried by viruses, COVID-19 has only about 14 genes. And to put that in perspective, you have about 25,000 genes. These green COVID genes are associated with the hijacking of the host cell's genetic machinery. So this is the hijacking part of it. And the rest of these genes contain information to make new bits and pieces that go into assembling the new viruses. All right, so that's a really quick primer on virus natural history. So viruses are pretty astounding and important, but they can be pretty scary parts of the natural world as well. Coronaviruses are associated with lots of diseases, and interestingly, they're associated with bats and other animals. And I'm going to turn this over now to Angie to tell you more about this.
Thank you, Professor Greene, for your descriptions and explanation about viruses and COVID-19. This is a great starting point in our understanding of the virus. Let's move to Professor Luis. Professor Luis, can you begin by telling us where COVID-19 came from?
Yeah, it's Lewis, Dr. Lewis. Hi, so where did this thing come from? So we know that the first handful of human cases were linked to a wet market in Wuhan, China. Wet markets are places where fresh meat, fish, produce are all sold, and these are really common in China and Southeast Asia, but also other parts of the world as well. Many of these wet markets have a mix of live and dead animals. It's a part of their culture in China to want to be able to see the specific animals that they're buying, and they want to see them slaughtered in front of them, so that they know that they're getting the freshest meat and they know what they're actually purchasing is what they want. So we go to a store, we pick up a slab of beef, a slab of meat, and it says lamb on it, and we just say, okay, we trust that that's lamb. But in their culture, they want to see it. They want to know what they're getting. And so that means that there are all these live and dead animals in a small space. And this is a great place for viruses to get transmitted among species. So I'm gonna see if I can share my screen with you for a second. If I can find it, here we go. So here is, has it come up yet? Nope, is it sharing the notes or the top part?
The notes for right now.
Yep, of course. Okay, hold on one sec, sorry. Yeah, you'd think that I didn't do this like for a living for the last couple of weeks. Ugh. Well, I'm having problems here. My computer's being slow. So I won't share the picture, but it's this picture that shows some ducks sitting right on top of a cage that has chickens in it. And you can see there's some nice poop that's coming from the ducks and being dropped on the chickens. So this is a great opportunity for viruses to be shared between species where in normal conditions, you wouldn't see this. And so there's a whole lot of different species that are interacting in these wet markets. Sorry, just one sec. So a lot of different species are interacting in these wet markets, but what also is happening is that you get the slaughter of animals right there. And so people want to see the slaughter, and the slaughter of those animals also means that there's a lot of blood. There's a lot of potential for aerosolizing all sorts of things. And so that's going to also increase the chance of transmission both between animals, but also from animals to humans. So the science suggests that this virus came from a bat, specifically horseshoe bats. And these are really cool animals. They're really interesting creatures. You should Google it. I had a picture, but it's too much of a problem to share right now. But they have these really interesting bits on their noses. They're called nose leafs. And it's thought that it's helpful for them to project sound when they're using echolocation. So they're really cool looking. You should look them up. So these are really interesting bats. They look like they are the reservoir of this disease. So what is a reservoir? It's a host that can maintain infection in the long term. So bats are the hosts of other coronaviruses which have jumped into humans, like SARS coronavirus. There was an outbreak in 2002 that also occurred in China, which also came from wet markets. And for that virus, there was an intermediate host that was important. And so we think that it transmitted, the infection started in bats, was transmitted to a civet cat in the wet market, and then the wet market, within the wet market, there was transmission from civet cats to humans. We don't know whether an intermediate host is important here for this disease. It's possible that it spilled over directly from bats, or maybe there's some unidentified intermediate host that's important from spreading from bats to humans here for this particular case. So this isn't a problem of just China. There is another closely related coronavirus that causes what we call MERS, and that's Middle East Respiratory Syndrome. And so that's caused several different outbreaks within the Middle East where bats spread infection to camels and then from camels to humans. The other types of viruses are being transmitted all the time from animals to humans all over the world. Another example is Hendra virus, which has caused 50 different outbreaks in the last 15 years in Australia. So it's passed from bats to horses and then horses to humans. Other diseases are spilling over from wildlife in the US. For example, I study hantavirus, which is an important zoonotic disease. So zoonotic means transferred from animals to humans. So this one's transmitted from deer mice to humans and can kill 1 in 3 infected humans. So these diseases that are passed from animals to humans are on the rise. Over the last 30 years, they've been increasing. And a lot of the outbreaks have been traced to changes in land use, agriculture, or wildlife hunting. So as human populations expand, we're changing the environment in ways that make these spillover events more likely to occur.
Great. Can you tell us more about how it spreads?
Sure. So unfortunately, this spreads really easily between people. It's more contagious than the flu, but it's transmitted in the same way that the flu is. The main route of transmission is what we call respiratory droplet. So what does that mean? So when we cough or sneeze, or even breathe, we're emitting water droplets. They're very small, and most of the time you only see them as humidity, like if you're blowing on a glass, for example. And most of them are large enough that they don't remain in the air, but they drop to surfaces. And so if you're infected and you cough, there's virus in those respiratory droplets that end up dropping to surfaces. So they don't remain in the air for very long, but are deposited on surfaces very easily. So people can get infected when they're nearby others that are coughing, or they touch surfaces that have been contaminated from someone else coughing around them, for example. So the main guidelines that you've been hearing about protecting yourself include staying at least 6 feet away from other people. And that's because these respiratory droplets don't tend to move that far. And so if you're at least 6 feet away, you're not likely to come into contact with somebody else's respiratory droplets. So you do have to worry, though, about surfaces. And so most of the respiratory droplets end up dropping to surfaces. And so we have to worry about the things that you're touching and all these different surfaces, and getting your hands contaminated potentially, and then touching your face, particularly your eyes, your nose, your mouth. That's how the virus can get in your body. So for example, if someone coughs near a table, the droplets drop onto the table, and now someone comes along and picks something, a pin up off of that table. They could potentially now have virus on their hands, and then if they don't wash their hands right away, then they could touch their face and become infected. And these, unfortunately, the virus is pretty stable on surfaces. So it depends on the surface, but it can last anywhere from a few hours to up to 3 days on some surfaces. So this is why it's really important to wash your hands. That's one of the things that's really been passing around. Anytime you touch something, you need to wash your hands. And washing your hands really does work. Like, some people are really into hand sanitizers and everything, but washing your hands really works because, as Erick explained, like so around the virus, on the outside of the virus is this envelope that's made of lipids. And that lipid envelope is basically, it's like its little protective bubble. And so if you can dissolve that bubble, then you kill the virus. So surfactants and everything that is in soap will destroy that protective bubble. And so that will kill the virus. You need to wash your hands for a good 20 seconds, get a good lather, and then fully rinse, but that will kill the virus. You don't need hand sanitizer or bleach, although those things do work. So this is why everybody keeps saying, wash your hands, wash your hands. So like I said, a lot of the particles are gonna drop to the ground or surfaces, but will they all? Can some of them remain in the air for a while? So this is a big debate actually right now, but most scientists agree that airborne transmission should be very unlikely. So you've probably been seeing changing recommendations about wearing masks. Initially, we were told that masks should only be used by healthcare workers and those that are sick. Healthcare workers are going to have people that are sick and coughing right in front of them. And so they really do need those masks to protect the people that are coughing right in their face. And so we really need to save the masks for them. And because when someone coughs in their face, those respiratory droplets can drop right in their mouths or in their nose. So the other recommendation is that sick people wear the masks. And that's important too, because if you're sick, then you're coughing directly into the mask, and those respiratory droplets aren't getting out and dropping to surfaces and exposing other people to that virus 3 days later. So this is why initially they had said it's not going to protect you all that much if you're uninfected, healthy, to wear a mask, because since there's not really likely a lot of airborne transmission, as long as you're staying 6 feet away from people, you don't need to worry about wearing a mask. And it can do harm if you're wearing a mask, but you're constantly adjusting it, because masks are really uncomfortable, and people that aren't used to wearing them often do kind of adjust them a lot, and then you're touching your face all the time, and that's the big problem. So the new recommendations that are coming out where they're like, well, maybe we should wear masks, everybody should. What that's about is that these recommendations are actually still more about protecting others from you than you from others. And so what I mean by that is there is a lot of asymptomatic transmission going on. So there are a lot of people that don't feel sick. They don't know they're sick, and they're walking around and they're contagious, and they could be spreading it. And so if they're wearing masks, they're stopping transmission to somebody else. And so this is the main reason why public health officials have really gone to advising people to wear masks who are healthy. It's really to stop the asymptomatic transmission. It's not to protect the people that are actually wearing them. Not that they can't be useful at all, but if you're gonna wear a mask, it needs to be a good, really thick mask, and you need to wear it properly and not adjust it a lot. So those are my main points.
Thank you, Professor Lewis, for sharing your knowledge and expertise about how this virus moves, where it started, and how to help us get better at protecting ourselves as well. I know there's been a lot of questions about whether or not masks work and how to use them correctly, so that's been incredibly helpful so far. Thank you so much. Let's move on to our last panelist for today before we open up for questions from the audience. Professor LaPier, can you share with us some of the misinformation you're seeing as it relates to COVID-19? There's a lot of conflicting information circling around about facts and what people think.
Yeah, so again, my name is Rosalyn, and I'm with the Environmental Studies Department at the University of Montana. And I am both a trained ethnobotanist. I was trained by Native women on the Blackfeet Reservation over many years. And I'm also an environmental historian. And in both of those kind of fields of study, I'm interested in looking at how human society interacts with the natural world. So with ethnobotany is sort of how does human society relate to and interact with plants, and what are those relationships that they have developed over time. And then with environmental history is sort of looking at why and how humans have interacted with the natural world, but also how the natural world has sort of interacted with humans. So for example, this semester I'm teaching a class on environmental health in Indigenous communities. And one of the things that we've looked at over the past semester is especially how diseases, epidemic and pandemic diseases have impacted Indigenous communities because many of the Indigenous people of North America are not immune to many of the diseases that came to what is now called the New World to North America. And because of that, Indigenous communities have suffered greatly. But it's something that a lot of scholars are now looking at and looking into. But as an ethnobotanist, one of the things I have been seeing, especially on social media, is that there is a lot of misinformation right now being shared about sort of treatments, preventions, cures for COVID-19 that are really based on kind of herbal remedies. And really, no one is immune to this virus. And so similar to Indigenous communities of the past that were not immune to many of the diseases that were brought over by Europe, we are now in a situation around the entire world where none of us are immune to this particular virus. And so we're kind of all in the same boat. And right now, again, on social media, I'm just seeing a lot of sort of misinformation about herbal cures that really are just not true. And as an ethnobotanist, ethnobotanists really take what we do very seriously. And so very similar to sort of a medical doctor, we would not prescribe or suggest particular treatments that we know do not work. And so when I'm saying that I'm seeing sort of that misinformation or even misleading information about how people can like boost their immune system or they can strengthen their lungs based on certain herbal remedies, there is no evidence right now, either in kind of the indigenous knowledge world or the Western science world, that shows that any of these types of remedies are, that they can either, again, kind of treat, prevent, or cure COVID-19. So I think that's all I'm going to share right now so that we can open it up for questions and conversation.
Great. Thank you, Rosalyn, for sharing that. I think all of us are excited to learn more about this when we dive deeper into how misinformation is affecting our communities, and your knowledge of histories and how infectious diseases can inform what we know now moving forward, and for how this affects how we're seeing information, especially spread on social media. So as Rosalyn said, it's time to open it up for questions from the audience. You're going to use the question and answer feature through your Zoom app to ask a question to our panelists. If your question is accepted, we will share it with the panel. As a reminder, we're not medical doctors and we will not be taking questions to diagnose symptoms. So we do have one question already. We're going to start with this one. This one came from actually somebody in the chat box a little bit earlier in the program. The question is, I've been reading that the virus can remain airborne for an hour or more. Is that a concern for virus spread? I think this question came while Professor Lewis was actually speaking. Professor Lewis, would you like to take this one?
Sure. There's really a lot of uncertainty about airborne transmission. So you might have seen, so there is some science to suggest that it could remain airborne for a while, for an hour or more. But those studies are actually pretty artificial conditions. Like they use this like drum type thing to get the droplets out, and it's not actually like a human cough. And so sometimes you need to be a little careful in how you interpret some of those studies. But basically what we're seeing now so far suggests that airborne transmission is not very important. There are some situations where it might be important, like in hospital settings, when you intubate patients and then you remove that intubation, for example, like there are certain conditions under which it might be easier to aerosolize and get that like tiny, fine little particles you would need to remain sustained like in the air. But we don't think right now that is very important for most transmission.
Great, thank you, Professor Lewis. Looks like we have another question from our co-host, Drew. What about viruses mutating? Are we concerned that COVID-19 could mutate? Who'd be the person to answer this one?
I guess I can go over that too.
Please, thank you, Dr. Lewis. Professor Lewis.
So viruses are mutating all the time. That's what they do. They evolve very quickly because they have such a short lifespan, and their replication machinery just is such that they don't do a great job making copies of themselves. And so there's just these constant little changes. So mutation's gonna happen. Like it already has happened. We've documented several changes in this thing over several months. So the question is, what are those mutations gonna do in the long run? And we can't really say now, but it's been transmitting for a while now, and the main routes of transmission haven't really changed much that we've seen. So really when we worry about mutation, probably for this type of virus, what we would need to worry about isn't it like changing its route of transmission or anything like that, because it's already super fit and as far as like it's getting transmitted really easily. When viruses mutate and then they get spread through the population, that happens when they're not as fit as they could be, and they could do a better job of getting transmitted. But this one's already doing a really good job of getting transmitted. And so it's not likely that the routes of transmission are going to change from mutation. So the things that we need to worry about are if we get infected and then recover, and then our immune system can now recognize it, the things that we need to worry about are, well, in a year or two, are we going to be able to be reinfected because it's mutated enough to be different enough that our immune system doesn't recognize it anymore? And that's something we're not really sure, but based on other closely related coronaviruses, we think that that's probably not likely that it will mutate enough to where our immune system won't recognize it, at least within the few years that it should take to get a vaccine.
Thanks, Angie. I'll just add a little bit more information. Yeah, I mentioned that coronaviruses are RNA viruses, and they have some of the fastest mutation rates of any of the 7 virus families. So as I'll just reinforce what Angie said, they are mutating and changing very quickly. And this is the reason, for example, that there's not just one influenza vaccine, right? What might have worked a year ago isn't going to necessarily work next year, because it will have had a year, the influenza virus will have had a year to mutate and recombine in weird ways. So that's why every single year there's a new cocktail of stuff for the influenza virus. So it's a great question, and it remains to be seen how much it changes in the future.
Yeah, the flu viruses are a little different in that there's so many strains circulating at one time, and there's a whole lot of different animal reservoirs. You can get recombination. And so the rate of mutation in flu viruses is likely to be a lot faster than what we're going to see for coronaviruses. But yeah, it definitely happens. Hopefully it'll be better in the flu in the sense that it'll take longer than a year or two for our immune system to not recognize it anymore. Whereas, like Eric said, with the flu, you really need to get that flu shot very often in order for it to be protective.
Great, thank you so much. So Rosalyn, would you like to share something about hand washing a little bit and the use of soap?
Yeah, I would just like to, this is a question that I get often asked by just people on the street. And I was actually asked this question yesterday. Somebody was asking me because they had run out of antibacterial hand wash, the pump wash. And they just sort of said, well, I ran out of this hand wash. Can I just use the same old, pardon my phrasing, hippie soap, right? And so this is what hippie soap looks like. Let's see if you can see it. Just a plain old bar of soap. And I just said, well, yeah, soap is thousands of years old. Human societies have been using soap for a very long time. It's basically made out of a couple of ingredients, right? It's made out of a fat or an oil and lye mixed together. And now today people add different types. If it's a hippie soap, they add usually some sort of plant ethnobotanical material to it as well, or some sort of essential oil to make it smell good. But basically all you're doing, as was explained earlier by Angela, you're just taking the soap, washing your hands, and it will slough off and either kill something, or you're just washing it away with the water. And so it doesn't matter the kind of soap. Bar soap, I think increasingly we're hearing from the research that's being done is much more effective than the liquid soap. And you can get any kind of soap out there, and whether it is vegan, which is an oil or a fat that is made from vegetable matter, or if it's non-vegan, which is a soap that's made out of an animal tallow or a fat. Usually it's made out of beef. Sometimes it's made out of other stuff. That these are, again, have been used by human societies for thousands of years, still very effective as a method of cleaning ourselves and keeping ourselves healthy and protected against these types of viruses. So.
Great, thank you. That means the soap I have in my bathroom is working. So that's some great validation. Thank you. Okay, we have our next question. What is it that makes this a novel, quote, novel virus? How is it so different from other viruses that we have never encountered this virus?
Is that one for me?
Sure.
Okay.
Take it away.
So novel just means it's something we haven't seen before. So we haven't seen this exact virus before. We've seen things that are closely related. So like I mentioned, the SARS outbreak from 2002, that's very closely related to this. And there's evidence that potentially those that actually were infected during that outbreak might have the antibodies that would cross-react, and so might actually be somewhat protective against this one. But that outbreak was much smaller. I think there was a total of about 8,000 cases total, and it didn't get too far outside of China. So novel, so what was the second part of the question? What does novel mean? And then what was the second part?
Sorry, I was on mute. The second part was, what makes this disease different from other diseases? Hold on, let me read it correctly. How is it so different from other viruses that we have never encountered this virus? So what makes the novel coronavirus significantly different from, say, other viruses that are similar to this one, but we haven't seen this one yet?
Yeah, so we have seen similar things. So the SARS outbreak from 2002, there have been several outbreaks in the Middle East called MERS. And both of those closely related viruses spilled over from bats into an intermediate host and then into humans. And so bats seem to be the host of a lot of different coronaviruses. There are pathogens in any species that you look for. So you could go and sample this squirrel that I can see outside my window right now, and you're gonna find some viruses. Are they gonna be important for humans? Most of the time not. But it's hard to tell just by looking and seeing that and sequencing a virus and saying, okay, this thing has a virus. So it's really hard to tell which ones are going to impact humans, but there's more research going on to try to do a better job of predicting, okay, when we see that there are all these viruses in this particular species, could any of them jump into humans? And so we're getting a little bit better at this. Is actually, like, people were predicting within the last few years that there would be a potential pandemic from coronaviruses of something we haven't seen before. So it's not maybe as unexpected as some people are making it out to be.
Great, thank you. And along those lines, we have another question here about how there's talk of the coronavirus being called a disease. Other viruses are not diseases. So what makes this virus a disease?
So a disease, technically, disease is symptoms. So there's a difference between the virus and the disease. So the virus is actually called SARS coronavirus 2, and the disease is called COVID-19. The disease is coronavirus disease 19 for 2019 when it first showed up. So it's the same difference between like HIV and AIDS. HIV is the virus and AIDS is the disease. And so that's what those terms mean. So the virus is the thing that causes the disease, but the disease is actually the range of symptoms that you experience.
Got it. Thanks for that clarification. That's really helpful. I don't think I completely understood the difference between the two either. So thank you. So Rosalyn, oh yes. Go ahead.
Just to let people know that there are some great kind of general audience resources that you can go to read about some of this. And just to provide sort of that longer historical piece, the Washington Post recently did an article on the kind of history of pandemics around the world over time, and talked a little bit about sort of, again, that difference between what's the difference between sort of just a small illness to an epidemic to a pandemic, and sort of discussed different diseases that have happened since ancient times to the present, and just explained a little bit about the differences, again, from anything from the Black Death during medieval times to our latest variations of the coronavirus.
Go ahead, Eric.
Yeah, that's great. And I just want to mention before I forget that we're assembling a list of resources on the Montana Natural History website And so Rosalyn, maybe you could email that link. There's already a bunch of resources up there. Some of them I just want to highlight. There's a bunch of great books for those of you who are interested. This is, I don't know, is that writing backwards? Yeah. So this is by Montana's very own David Quammen, who lives in Bozeman. He's a fantastic author, science writer. And this is called Spillover, Animal Infections and the Next Human Pandemic. And so he spent a lot of time going all around the world talking to epidemiologists and scientists. So this is a great book for a public audience that gives you a lot of just general information about viruses and viral diseases and pandemics. And this is not a surprise. People, epidemiologists, scientists, the Department of Defense, public health people have been predicting for a good while that we're going to have a pandemic. It was probably going to be a coronavirus. It would crash the economy, and we were not ready for it. So that's one book. There's another good one called The Viral Storm. And these links are on the webpage. This is by Nathan Wolf, who's a really great virologist, and he goes all around the world looking for viruses and hotspots for emerging infectious viral diseases. And then this is a great little book. This is the thinnest, the thinnest of the bunch. This is by Carl Zimmer, who's a fantastic science writer. It's called Planet of Viruses. And the first two are a little sobering and scary because they're all about pandemics. This has some stuff about pandemics, but it's a lot more about the basic natural history and biology of viruses. So I just want to get that out there. These are three books that I'd really recommend for those of you who are interested in just delving into this important and fascinating topic.
Thank you, Eric, for those great resources. As Eric said, those resources will be posted on our website. Our co-host Drew just pasted the link of that into our chat box. Before we finish though, we have a handful of more questions we'd like to try and get to. And a couple of them that have come up in our Q&A through the Zoom are directed for Rosalyn. So Rosalyn, one of these questions is, what analogies do you see, if any, between how past diseases have affected Native Americans and how COVID-19 is affecting or might affect Native American groups?
So right now in most Native communities, we're kind of in the calm before the storm. Really the only Native community that's been really seriously impacted right now by the COVID-19 is the Navajo community, and the Navajo reservation right now actually has more cases of COVID-19 and more deaths than the state of Montana, for example. And the state of Montana, we still are in that kind of calm before the storm phase. And so Native communities, excuse me, are preparing, as are a lot of communities, for the time that this disease is going to come within our community. One of the things I'd say that I had mentioned sort of earlier is, as with former times, there is no immunity to this. And the difference is, in previous times when Europeans were bringing diseases to North America, Europeans had some immunity to various illnesses. In this particular case, nobody has an immunity to this particular illness. It doesn't matter who you are. And this is something that, again, in terms of misinformation that I'm seeing out there, it doesn't matter if you are rich or you're poor. It doesn't matter, if you're tall or you're short, if you're skinny or you're fat. It doesn't, none of those things matter. None of us are immune to this. And unfortunately, one of the things that I think that we will probably see, and we are kind of seeing this already with the African American community, is that those communities that already have serious health disparities and health issues are going to be the ones that will be impacted greatly. And we're already seeing this as well in terms of populations, right? So the older population that has more health issues is the population that is being impacted more greatly. I think those other populations, ethnic communities that have higher health issues are also going to be impacted more than other communities. But in terms of the actual immunity, no one's immune to this, and it's going to impact everybody. And it just sort of depends. Those of us in the center of the United States, it's sort of heading our way at this point. It is here in Montana and the Great Plains, but it hasn't impacted us as much as it has in other communities quite yet.
Great, thank you. We have another question for Dr. Lewis. So you spoke earlier of how long this novel coronavirus can persist on surfaces. What does this mean for food safety?
Yeah, that's an open question, but people seem to think that it's not, that food safety isn't really an issue. So there hasn't been any documented instances of transmission from food. So people are assuming, so we know, so I mentioned soap is really important in killing the virus, but heat is also really important. So if your food is warm enough, then that's not a problem at all. If you're talking about some other types of food, you know, that's not heated, then you might have to worry a bit, but there's really not been any documented cases of transmission from food. So I don't think it's something you need to be really worried about. So what people have been suggesting is, like, you know, when you get takeout food, maybe change the container over to your own container and then wash your hands. So it's more likely that there might be virus on the containers than actually in your food.
Great, thank you for that clarification. We've got a couple of questions about vaccines next. So I'm thinking about, how long does it take to make a vaccine, and how will vaccines relate to lessening the impact of COVID-19, and why does it take so long to make them?
So vaccines, like in the past, it's been at least more than 7 years to go from starting development to an actual usable vaccine that could be distributed in the public. Things are really rushed right now, and there's a lot of there are multiple vaccines already in development. And that is already like a really huge push beyond what's happened over the last 10 years. So that's really good news, that there might be a vaccine available within a few years. So part of the reason why it takes so long is just to figure out, like, what is the best target? Like, how do I figure out what little bit to put in the vaccine that would be protective? So I have to stimulate an immune response that is close enough, and it's going to recognize this other thing that I'm going to introduce. So it's really tough to try to figure out exactly what do I put in a vaccine in order to be protective against this thing. But we're getting better at that. And that's why things are faster now than they used to be. But even if you know exactly what to put in your vaccine, you still need to do clinical trials to prove, well, first you usually use animal models, and then you actually use it in humans, just to make sure that it's fully safe, because we don't want to put this thing out there and expose all these people to a vaccine that might not be perfectly safe. And so that's why that's going to be the longest part of this, is just to make sure that it's safe in animals and then that it's safe in humans before it can be distributed really broadly. But a vaccine is going to be really important here. So I'm going to talk next week about general epidemiology, like what do outbreaks usually look like? We're showing like the epidemic curve, what causes that, how do we flatten the curve, and what does that mean? But what we're doing right now is trying to flatten that curve and get rid of infection. But we can't completely eradicate it here, because it's always going to be introduced from somewhere else. Like, if we don't eradicate it in all of the world, there's chance for it being re-imported here. And so a vaccine is going to be really important to really eliminate this problem. But that is going to be in the longer term. So I'm going to talk about these kinds of issues a bit more next week.
Great, thank you. We've got a few more questions, and a couple of them are pretty similar. I'm going to start with a kind of a scientific question and Maybe you can also expand on this a little bit more as well, but is there a suspected R naught value for corona? And can you also explain what R naught means as well for our audience that might not know what that is, myself included?
Yes, so that's actually what all my next webinar is going to be about. So tune in a week.
Perfect. Great.
I can just give the broad definition, or should we just wait and get to a different question?
Well, we only have about 5 minutes left,
Okay,
so let's save that one for next week, and we'll definitely be sure to cover that. We do have a couple questions about more of a long-term or global presence and what this is going to mean. So what we have here is, what sort of long-term changes do you think we on global and personal levels need to make to prevent future pandemics or better control their spread? And in time, will it be possible to develop a level of global immunity to a virus like this?
So what, give me the first part of the question again, so I can.
I'll just say, you know, if the past is any help, I would sort of answer that question as sort of no. That there are going to continue to be these types of pandemics that will continue to occur, and it just depends on, as we've seen in the past, the ones that Angela has mentioned already, like MERS, that did not become sort of a huge global pandemic the way this has become, and that there are some of these viruses that live a certain sort of lifespan and then peter out. But we've seen this happen again throughout the years of kind of human, you know, the life of human society.
Eric, go ahead.
I'll just jump in and just add a few things and let Angie handle this as well. I think one of the major things that we're going to see, not only in the United States, but probably around the world, is people are realizing now that we have to be much better prepared in terms of testing and public health tracking. So some of the most effective ways of keeping viral diseases from turning into these global conflagrations and pandemics is if we have much better testing. So, and then once you have good testing and know who actually has it, especially in these cases that, like Angie was mentioning, you know, a lot of people are asymptomatic. They don't know they're sick, but they're infecting others. So testing is really, really important. And as a country, we've been really caught unawares. And so we're playing catch up right now. So as are many other countries. So I think globally, I think politicians are going to realize that we need to be much better prepared in terms of being ready to test and do public health tracking, and also just things like having stockpiles of gloves and sanitizers and ventilators and whatever. So I think that's going to be a major, major shift in thinking we're going to see that's come out of this.
Yeah, I'd just reiterate the testing is a huge, huge part. So this is going to continue to occur. So I mentioned that these zoonotic disease outbreaks are on the increase. So things have only been increasing over time. We're seeing more spillovers from animals to humans, and that's because we're changing the environment and making it more likely to occur. And so this is only going to increase unless we do something drastic as far as stopping contact with wildlife and conserving biodiversity and a lot of the things that are leading to these spillovers. But given that we know this is going to continue to happen, big things are like being more prepared than we were this time. And the testing and then contact tracing, if you're really good about that, if you test as many people as possible, and then every infected case, you talk to who were they in contact, who were they in contact with, and you create this big ring around all your infections, then you really can do a good job of containing things. And that has happened with outbreaks in the past.
Great, thank you. And we're going to continue to dive more into questions about what this is going to mean moving forward and what it means on a global scale. And I would like to come back to that question on the possibility of global immunity moving forward into the future during future webinars that we hold in this series. We are at about 4 o'clock though, so I want to take a moment here to just thank all of our viewers today. We had some really great questions come in, and we're going to continue this discussion next Friday at 3 p.m. Mountain Standard Time, 2 p.m. Pacific Time. We'll be jumping into epidemiology with Dr. Lewis, Professor Lewis, and what it means to flatten the curve. Thank you to all of our panelists here today, the Montana Natural History Center and the University of Montana for this partnership in our new webinar series, The Age of the Coronapocene. I hope everybody has a safe and fun, socially distanced weekend, and we will see you all next week. If folks have questions that they didn't get answered and would like to reach out to any of the panelists or the Montana Natural History Center, I can help make sure those questions get to the right people. I'm going to put my email address in the chat box. Feel free to shoot me an email if you'd like to get in touch with any of our panelists or follow up further. Otherwise, this ends our webinar for today, and we will see you all next week.